hopefully
brilliant. We can see your screen now. Yeah, we can.
Cool. And I've also got, just going to make sure I've got the chat function open. So if anybody is by a computer and wants to type in any questions, feel free to do so, or else I'll take questions at the end. So this presentation kind of follows on and slightly overlaps with the last one for those of you who came to the last presentation on immunotherapies. But this presentation is more widely about the use of antibodies as cancer treatments, partly because there's a lot of stuff changing and because they're used in quite diverse ways to achieve different effects, partly because I think there's some confusion as well. I know it's always really hard to fit cancer treatments neatly into categories. I was just looking at the Macmillan website the other day and their learning hub, and they sort of they separate out antibody-based therapies and immunotherapies, and of course there's overlap because some antibodies are used as immunotherapies and it can get rather confusing. So I'm hoping this presentation will dispel some of that confusion and help bring some clarity. So I'm going to start with some basic information about what antibodies are and why we should use them as cancer treatments. Then I will look at some of the improvements and innovations around the use of antibodies as cancer treatments. There's a lot happening with the antibody-drug conjugates. There's a lot happening with the T-cell engagers, and there's lots happening with CAR T cells and TCR-engineered T cells. So those are the three main groups that I'll be looking at. There's some other bits and pieces that I slot in along the way. What I'm not doing this evening is talking about checkpoint inhibitor immunotherapies. I talked about them a lot in the previous presentation. They are, of course, at the moment, all antibody treatments. So they would fit into this had I not already described them in a previous presentation. So if you didn't, if you weren't attending that last presentation on immunotherapies, please do check that out if you would like to know more about the checkpoint inhibitors. I just ran out of space in this presentation and decided that I didn't have room for them this evening. So I will begin with what are antibodies and why we use them as cancer treatments. And I'll start with the fact that antibodies are proteins. They are large proteins. They're made just by one set of immune cells of our immune system, and that's the B lymphocytes, B cells. B cells produce them and release them into the bloodstream as part of our body's defense against infection. They are incredibly precise in what they attach to, which is very useful, and they trigger an immune response against whatever it is that they've become stuck to. Anything that an antibody might stick to is known as an antigen. I'm so sorry, I can hear my dog. It might be because my husband has come home. If she doesn't stop, I will dash.
For those of us that know, this happens every time at this hour. But it's quite entertaining. She was. And she will be quiet in a moment. Don't worry, Elaine.
Yes, if she doesn't calm down in a second, I will go and get her many treats and help her relax. So that anything that an antibody attaches to is known as an antigen. And when an antibody attaches to an antigen, it then triggers an immune response against it. Antibody-based cancer treatments are similar to the natural antibodies made by our B cells, but they have been deliberately designed so that they attach to a carefully selected target on cancer cells. Generally, it's something on cancer cells. With the immune checkpoint inhibitors, it's something on T cells, but whatever it is, it has been carefully selected as being that antibody's target. Now, in order to manufacture antibody treatments, you can't do it just in a lab. You can't ask a chemist to construct an antibody piece by piece. It is too complicated. There's thousands of separate molecules all to stuck together to create that antibody structure. I'm going to give her treats. If you would mind, excuse me just for a second. I will switch my camera and microphone off and I will come back in a second, armed with many treats to keep my lovely little dog quiet. Just one moment.
Sorry, and just as I then release my mic, I then start a coughing fit. So anyway, sorry about that. Sam, is that you who I know from before? Have I got the right Sam Bird? Or is she quiet?
No, I don't think I've met you before.
No, I don't recognize your voice. So no, it's I know a Sam Bird then from another life. So that's fine. Welcome anyway. Where do you work? Sorry to put you on.
I work at SACT Mandeville.
All right. Well, welcome to the session.
Thank you.
I hope you all find this evening, like I love these sessions that are giving us the science behind all these drugs, and especially like with our trastuzumab. Welcome back, Elaine.
She has been fed chewy treats in the hopes of calmness. Sorry, did you want to carry on in what you were saying?
I was just trying to fill in the gap. That's all, the silence. So you start with the slide as at the beginning is probably easier for you, Elaine.
I'm still sharing, aren't I?
You are? Yes.
Yeah, perfect. Okay, so I was just making the point that antibodies, because they're very complicated proteins, you can't ask a chemist to make one. They're too complicated. So what you do is you work out the antibody you want, you work out the genetic instructions, the gene for making that antibody, the strand of DNA that would tell a cell how to make that antibody, and you put that DNA into manufacturing cells and you grow those cells in vats, and they churn out billions of identical copies of the antibody you want, and you purify them and you give them to patients as your treatment. Because you're using living cells to manufacture them, they are a form of biological therapy. That's in its, the meaning of biological therapy is a treatment that's been manufactured using living cells. Now, it gets used sometimes more widely than that. It gets used as a sort of catch-all term, but that is the definition of a biological therapy. And they're called monoclonal antibodies because you've genetically modified these manufacturing cells in exactly the same way. So you've got billions of cells all containing the gene for the antibody that you want, all manufacturing the antibody that you want, that you then purify. So you've got billions of copies of the exact same antibody made by a single clone of genetically identical cells that all contain the gene for making the antibody that you want. So monoclonal, one clone of genetically modified cells is manufacturing that antibody for you. So that's why they're called monoclonal antibody therapies. And the ending of the name of a monoclonal antibody will always be MAB, standing for monoclonal antibody. So the structure of an antibody is like this. It's sort of a Y shape. The two orange tips are what the antibody uses to attach to its target. They're the antigen binding sites. The antigen binding sites just refer to the very, very tips, and they are part of a slightly larger region of the antibody known as the variable region because it varies from one antibody to the next. The constant region is constant between antibodies, and so it's called the FC, the constant region. So the tips attach to the target. The back end, the FC region, is what recruits and activates white blood cells. And that back end also determines what happens to that immune system in your body. Sorry, what happens to that antibody in your body. Antibodies tend to stick around. You know, if you've had COVID in the past 2 or 3 months, you've still got antibodies to COVID proteins floating around in your bloodstream. They don't immediately get destroyed in the way that a lot of small molecule cancer treatments would. The small molecules, you give a daily tablet, a twice daily tablet. A lot of these antibody therapies you can give as a 2-weekly injection, a 3-weekly injection, a monthly injection even, because the level of antibody in the body stays pretty constant. It takes a long time for that to be destroyed. And they are versatile. You can decide what they attach to. You can tweak the back end of antibodies. You can alter the order of the amino acids that they are made from. You can add complicated sugar molecules. All of these changes tweak the ability of that antibody to attract in and activate white blood cells and create an immune response. You can also attach chemotherapy to an antibody, and you can use the antibody as a delivery device to take that chemotherapy to the patient's cancer cells. You can create mix and match proteins, antibody-based proteins that have new properties, tethering together the tips of different antibodies and creating different arrangements with new properties that the parent antibodies wouldn't have had. And if you want, you can purify some of the patient's own white blood cells and put in the gene for an antibody-based protein, and those white blood cells will then make that antibody-based protein of your choice, such as a CAR protein, a chimeric antigen receptor protein, which is part antibody, and that's CAR T cell therapy. So you can do a lot with the basic structure of an antibody, tweaking it, rearranging it, changing it, forcing the person's own white blood cells to make it. So there's a lot of possibilities with these antibody-based cancer treatments. Now, one of the sort of more confusing things about antibodies is depending on how they've been made and depending on what they target, we might classify them as a targeted therapy, or we might classify them as an immunotherapy. And the dividing line between the two shifts around depending on who you're talking to, what their background is, what treatments they're giving to patients, what patients they meet, what level of understanding they have of the treatment they're giving. That dividing line will shift around. So this is my division of how I separate them out. So I would say a conjugated antibody is a targeted therapy. You're using the antibody to target the patient's target the patient's cancer cells and deliver chemotherapy directly to those cancer cells and kill them outright. At the other end of the scale, you have treatments like the checkpoint inhibitors, the T cell engagers, the CAR T cells. These wouldn't work if the patient's own white blood cells weren't able to join in the party. The checkpoint inhibitors, you are boosting the activity of the patient's own T cells. The T cell engagers, you are drawing in the patient's own T cells. CAR T cells, you are modifying the patient's own T cells. So you are relying on the person's own T cells as playing a role and being engaged and activated by your antibodies, but it all relies on the person's own T cells of their immune system. And then the ones that are possibly more difficult to categorize, things like the antibodies given to some people with solid tumors, where the point of that antibody, the purpose of their antibody, is to kill cancer cells outright by blocking a protein on their surface that they were relying on for survival, such as the EGF receptor in people with bowel cancer, HER2 in some people with breast cancer or stomach cancer. The cancer cells have these proteins on their surface. You block the actions of those proteins, the cancer cells die. However, these are still antibody-based treatments. Cetuximab, trastuzumab, they're still antibodies. You're hoping they will kill cancer cells outright, but you really don't mind if they start attracting in some useful white blood cells and activating them and for them to join in. And for some patients, that might be a major part of their activity. Some patients, it doesn't happen at all. So whether you class them as some form of immunotherapy or not depends on who you are and how you look upon this process. Angiogenesis inhibitors, I've put in grey, I've put a question mark. There are some theories as to how angiogenesis inhibitors might engage the immune system or might affect the immune system. But by and large, we think they work independently of the person's immune system. The antibodies that attach to proteins on hematological cancer cells, like the protein CD20 and the antibody rituximab, well, they are immunotherapies in that they attract in and activate white blood cells, and that's how you kill cancer cells. But they are targeted in that they are targeting a protein on the surface of the patient's cancer cells. So you could easily argue for them to be a targeted therapy or an immunotherapy. I call them an immunotherapy, as do many other people, because they work by engaging the patient's immune cells. But it is a dividing line that is not clear-cut, that it does depend on your background and your the treatments you work with and your familiarity with the range of antibody-based treatments as to how you classify them. One way that scientists have aimed to improve upon a simple naked antibody is to attach a payload to the antibody and use that antibody to deliver chemotherapy. You can add multiple copies of a chemotherapy drug to an antibody without really changing its size. Antibodies are enormous compared to the size of a chemotherapy drug. So you can afford to attach lots of copies of a chemotherapy. Your goal is for that antibody-drug conjugate, antibody with the drug attached, you hope that it will attach to a protein on the surface of the patient's cancer cells. Hopefully what then happens is the membrane folds inwards and the antibody still attached to its target ends up inside the cell. The drug, if you've designed it to do this, the drug then gets released. Hopefully that drug is very toxic and it kills the cell outright. But this is an antibody that you're using, and it still does have the potential to kill cancer cells outright by merely blocking its target or by attracting in and activating white blood cells. So you're just adding another layer to this antibody's possibilities and its mechanism of action. These antibodies can still attract white blood cells, can kill cancer cells outright of their own accord, but you've added this extra dimension of using them to deliver chemotherapy. Sorry about the dog. The antibody-drug conjugates then have these three main elements. You have the antibody, you have the chemotherapy, and you have the linker holding the chemotherapy to the antibody. And all three of these things have been going through lots of changes and innovations as time has gone by. The antibodies, we're trying to switch to humanized or fully human antibodies so that they're less likely to cause infusion reactions. We're switching away from non-cleavable linkers to cleavable linkers. Cleavable linkers mean that you end up with more drug released inside the person's cancer cells. And the drug that's getting used, we keep changing our minds as to which drugs are the most useful, but you have to balance out potency versus toxicity. You do really need very potent forms of chemotherapy because you're not ever going to get that much of it delivered to the person's cancer cells. So it needs to be potent. But if the linker is not entirely stable and is liable to break within the bloodstream, then you're going to get toxicity if that drug is very, very potent. So you balance out the fact that, of course, you're going to get some leakage. You're going to get some of that drug leaking into the bloodstream. But you've got to balance that with the fact that you're not ever going to get that much delivered to the cancer cells, and it needs to be potent enough to kill them. So scientists are trying to design antibody-drug conjugates that cause fewer infusion reactions, where the drug is a soluble drug. And this is important because if it's soluble, then it gets released inside cancer cells and it leaks out into its environment. And so you get a bystander effect of nearby cells being killed as well. Linkers are getting more stable, so you get less of the drug being released in the bloodstream, but are more breakable inside cancer cells in the environment within a cancer cell. And then lastly, scientists are working out ways to be more precise and more consistent in the way the drug gets linked to the antibody, making this more uniform, more consistent, making the drug so that it attaches only to particular places on the antibody, exactly where you want it. So these drugs are getting better all the time. And I thought a good comparison is trastuzumab emtansine versus trastuzumab deruxtecan. And I thought I would compare them side by side. So trastuzumab emtansine is Kadcyla. Trastuzumab deruxtecan is Enhertu, if you're used to the brand names. Trastuzumab emtansine is the older treatment. The leaker is non-cleavable. You've got 4 or so copies of the drug rather than 8. Emtansine has quite a long half-life, which can add to its toxicity. It gets trapped inside cells, it doesn't leak out. Sorry, my husband's just come home, so she's going a bit. I'm so sorry. She goes absolutely nuts whenever my husband comes home.
That's okay.
Some days. Trastuzumab deruxtecan is actually changing the way that we treat breast cancer. It's changing the way we treat other cancers as well, because with trastuzumab emtansine, you need a lot of HER2 to be present on the cancer cells in order for it to work. So what you classify as HER2 positive in breast cancer is actually lots of HER2. Whereas trastuzumab deruxtecan, because of these better properties, it will work against cancer cells which have much lower amounts of HER2 on their surface. And this is something that NICE is currently grappling with. They're due to make a decision in November about expanding the number of women with breast cancer who we classify as HER2 positive. I'm saying, okay, well, there's the really high HER2 that we've always classified as HER2 positive, but there's also the HER2 low breast cancers. And about 60% of breast cancers are HER2 high or HER2 low, rather than the 20% that's HER2 high. So if NICE decides that yes, trastuzumab deruxtecan can be given to people with HER2 low breast cancer, suddenly we're giving it to a much larger number of patients with breast cancer than we have done previously. And there's also many trials of trastuzumab deruxtecan in HER2 high and HER2 low stomach cancer, gastroesophageal junction cancer, ovarian cancer, cervical cancer. There's lots of cancers where you don't necessarily get the really high levels of HER2 that you might see in breast cancer, but you do see some HER2, and it might be that trastuzumab deruxtecan is a decent treatment option for those patients. This is a list of the currently approved antibody-drug conjugates. A lot of them are licensed for people with the hematological cancers, but this is increasing all the time. You've now got sacituzumab govitecan for triple-negative breast cancer. It targets a protein called TROP2. Enfortumab vedotin targets a protein called Nectin-4, and so on. And there's a lot of these in trials. There's a lot of them coming through. So I'm sure this list will get longer. Now moving on to our use of antibodies as immunotherapies and the sorts of innovations that are happening in this area. And as I said, obviously you could include checkpoint inhibitors in this list of antibodies that act as immunotherapies, but I'm not talking about the checkpoint inhibitors this evening. What I will talk about briefly are the naked antibodies that attract in white blood cells. I then spend more time on the T-cell engagers and the CAR T cells and the innovations there. So the first group of treatments are the naked antibodies, the simple antibodies, straightforward antibodies that attach to a target of your choice, such as CD20 found on the surface of the B-cell cancers. These antibodies, they have their intact back end. So like natural antibodies of our immune system, they can attract in natural killer cells. They can attract in macrophages. They can also act as an attachment point for complement proteins that float around in our bloodstream and do all sorts of stuff for our immune system. There's a set of complement proteins that can create the membrane attack complex, punch holes through the surface of the cell and make them die. So these are pretty straightforward, plain naked antibodies, they're called, that attach to a target and attract in white blood cells and complement proteins. And I've listed just a few of these antibodies here, predominantly used as treatments for hematological cancers, because those are the cancers where there are some active and useful white blood cells in the vicinity of the cancer cells. Solid tumors, this approach doesn't work. The natural killer cells, the macrophages in a solid tumor, they're there, but they've often been changed by the cancer cells so that they're less active, they're less useful. You might be able to recruit a few of them, but they're going to struggle to kill cancer cells. So it's a much more effective treatment strategy against the hematological cancers than the solid tumors. You can improve on these basic, simple antibodies. You can, as I've said before, make lots of subtle tweaks and changes to the back end of the antibody, the part that attracts in white blood cells and complement proteins. The ones where you alter the sugar molecules that get attached, these are the glyco-engineered antibodies. Obinutuzumab is an example of them, but there's lots of the antibody treatments being created now. They do incorporate some of these tweaks and changes to try and enhance the recruitment of white blood cells. But the T-cell engagers, the antibody-based treatments that physically connect together cancer cells to T cells, this is a much bigger sphere of innovation at the moment. The T-cell engagers are mix and match proteins. They would not exist in nature. They would never be normally produced by any cell, but you can design them and you can use cells to manufacture them. Where this approach started was with a treatment called blinatumomab, which is a treatment for people with acute lymphoblastic leukemia. Blinatumomab is one of these T-cell engagers, bispecific T-cell engager. Bispecific because you've taken the two tips of two different antibodies, so they have two different antigen binding sites taken from these two different antibodies. A T-cell engager because one end of that novel protein now attaches to a target on T cells and draws them in. The other end attaches to a target on cancer cells. So their structure is that you've got two SCFVs. So this is an SCFV is the tip of an antibody, but it has been constructed using a little linker to hold the whole thing together. A normal antibody, the two tips are held in place by the rest of the antibody structure. If you just created these two tips, they would float free and separate. You have to link them together. So you do that with a little piece of linker protein to connect the whole thing together, and now it's called an SCFV. So you've got two SCFVs held back to back by a linker to create that bispecific T-cell engager. Now, the bispecific T-cell engager, I've already mentioned, is blinatumomab. This is the one that is used as a treatment for acute lymphoblastic leukemia. One end attaches to CD3, a protein very reliably present on the surface of T cells because CD3 is part of the complex of proteins known as the T-cell receptor complex. The other end attaches to a protein called CD19, reliably present on the cells of acute lymphoblastic leukemia. So you're creating a bridge, a physical link between the person's cancer cells and the T cells of their immune system. And this engages the T cells and tells the T cells, kill your target, kill the thing you've now become tethered to, kill that cancer cell, hopefully. Blinatumomab is a useful treatment. It is increasingly given to people with acute lymphoblastic leukemia in a number of different disease stages. But one of the big limitations with blinatumomab is it has a very short half-life in the body. It lasts not very long at all. So 2 hours after administration, half of it's already gone. That's because there's no back end. You know, you've taken the two tips of two different antibodies. There's no FC region, and it's the FC region that tells the body not to destroy it. Without an FC region, this structure, this protein, gets destroyed very quickly. and it's destroyed so quickly that you have to administer the bispecific T cell engager using a continuous infusion using a portable pump. Not convenient for the patient. Always the possibility that the tube into the body will get infected, it's an open wound. So it's not ideal. And that has been leading to innovations and improvements to create T cell engagers with a longer half-life. So blinatumomab and other structures, similar structures, don't have that FC region. But there's a whole range of new structures being created that do incorporate a modified FC region. It's modified because the whole thing would get too messy and too confusing if that FC region were attracting in macrophages and complement proteins and natural killer cells. You don't want that. That's just too confusing, too messy. The T cells would struggle to gain access. So it's a modified FC region. So it doesn't attract macrophages, natural killer cells, and complement proteins, but does tell the liver and kidneys, don't destroy me. So you get the long half-life of an antibody without too much confusion. What is a little bit confusing at the moment, though, is, as ever with these things, the language and the way we talk about these treatments evolves and struggles to keep up. So we have been used to the idea that this is blinatumomab and this is a bispecific T cell engager. However, all of these other structures, to some people, they also get called bispecific T cell engagers. They do have two specificities. They do engage T cells. Other people, though, call the blinatumomab-like structures the bispecific T cell engagers, and everything else is just a T cell engager, or it's a bispecific antibody. So the language hasn't caught up yet. And if somebody talks about a bispecific T cell engager or a T cell engager, you would have to ask them, what is the structure? What are you talking about? You may be able to guess by the way it's administered. If it's given as a continuous pump, then it's probably something similar to blinatumomab. If it's given every 3 weeks, then it's probably one of these other structures with an FC region. So you might be able to guess. The two structures where we've got the most treatments in trials or approved for use are the two I've circled here, and I've called them A and B so that this table makes sense. So I've listed all the T cell engagers that have been approved and some of the ones that have not yet been approved. So the ones that have been approved: mosunetuzumab, epcoritamab, glofitamab, teclistamab, elranatamab, and talquetamab. Now, as yet, none of these has been NICE approved, and that's because of differences in the threshold of the amount of evidence needed to persuade NICE versus the FDA or even the EMA. The trials that have led to these FDA and EMA approvals are single-arm trials. There was no comparison. Patients were given these treatments. People said, oh, yes, looks like it's working. They gathered together all the survival data on the people given the treatment, and they presented that to the regulator. NICE, when it rejected mosunetuzumab earlier this year, it said, look, we just don't know what to do. We don't know what the level of benefit is. Single-arm trials are notorious for being misleading. You can, if you've got a patient who's been through lots of treatments, who's very, very poorly and likely to die soon, you might choose not to put them forward for a clinical trial because you don't want to put them through that, and really they're terribly ill and they wouldn't be eligible for that trial anyway. If you've got a patient who has been through a lot of treatments, their disease is resistant to all of them, but they're relatively young, they're relatively fit, they're relatively healthy, you might put them forward to a clinical trial. They are more likely to benefit from whatever treatment they're given than somebody who's in a very poorly state, who has lots and lots of other comorbidities. So I guess the point I'm making is these single arm trials, the the data can be skewed by the fact that it tends to be the fitter patients with slower progressing disease who get entered into these trials. So NICE is saying, look, you know, we've seen your results. It looks promising. It looks exciting, but we can't tell what level of benefit these treatments are given when it's only single arm trials that we're seeing. There's no comparison arm. How do we calculate their cost effectiveness if there's no comparison arm in that trial? So NICE so far hasn't approved any of these treatments, and it's difficult for them to calculate their cost effectiveness until you've reached the point of having some comparison data there with whatever would be the standard treatment or supportive care, and so you can then work out whether it's worth the NHS providing it or not. So it might be a little while before we see NICE approvals. The data does look exciting. It does look like these treatments can work for patients, but exactly what level of benefit they give and how that's different from other treatments, we've yet to find out. One other improvement or innovation with these T-cell engagers is to say, well, instead of fusing together the tips of two different antibodies, let's fuse together, link together the tip of an antibody with the tip of a T-cell receptor. This opens up the range of potential targets, and it massively increases the range of potential targets, and I'll explain why in a second. So I've already mentioned antibodies. They have two antigen binding sites, two tips that attach to a target, and that target is generally a small region known as an epitope of a much bigger intact protein found on the surface of a cell or on a bacteria or on a virus. They attach to a small part of a much bigger thing. T-cell receptors that sit on the surface of T cells have a lot in common with antibodies. A lot of their structure is the same, and probably going back in time in evolutionary terms, probably they started out as a single thing that's then been copied and diversified, and B cells have become different from T cells, and the T cell receptor and antibodies are two different but related proteins. T cell receptors stick out from the surface of T cells, and they have one antigen binding site rather than two antigen binding sites. And crucially, the antigen for a T cell receptor is different from an antigen for an antibody. The antigen for an antibody is an intact protein. The antigen for a T cell receptor is a tiny fragment of a protein known as a peptide, displayed by an MHC protein. All our cells are covered in thousands of these little tiny cup structures that we call the MHC proteins, and nestled inside each MHC protein is one of these potential peptide antigens for a T cell receptor. Potentially it's something that a T cell could attach to. Crucially, these peptide antigens come from proteins inside the cell rather than being things naturally found on the surface of a cell. An antibody can only ever attach to something naturally found on the surface of a cell. There's only so many things found on the surface of cells, so there's only so many potential targets for an antibody-based treatment. But a T cell receptor attaches to tiny fragments of proteins that have come from internal sources. And so suddenly there's a whole world of possible antigens for a T cell receptor based cancer treatment, a much bigger range of possibilities of what you design that T cell receptor based treatment to attach to. One added piece of detail, though, with treatments based on the tips of T cell receptors is that you have to be more careful in selecting your patients for these treatments, because the T cell receptor naturally connects with its target, a peptide antigen sitting inside an MHC protein, but the T cell receptor is also making connections with the MHC protein. And so a treatment that is based on the structure of a T cell receptor rather than an antibody, you've got to do HLA matching with the patient, because all of us have different shaped MHC proteins, and the T cell receptors can only attach to peptide antigens presented to them by particular MHC proteins. So you've got to check that the patient has the right MHC proteins in order to prescribe them your T cell receptor based treatment. The T cell receptor based treatments that are the most advanced at the moment are the ImmTACs, is what they are called. And instead of being a bispecific T cell engager made of a back-to-back fusion of the tips of two antibodies, you've got a back-to-back fusion of the tip of an antibody that attaches to CD3 on T cells, and then you've got a soluble version of a T cell receptor that attaches to whatever you've decided is your target on the patient's cancer cells. So the target of the T cell receptor bit is a peptide antigen presented by an MHC protein on the person's cancer cells. The antibody bit attaches to CD3 on the patient's T cells. It does the same job as a bispecific T cell engager or a bispecific antibody, that it creates that physical link, but you have a much, much bigger range of possible targets when you use this structure incorporating the tip of a T cell receptor. The first one to be licensed as a cancer treatment is called tebentafusp. This is a treatment for uveal melanoma that occurs in the eye, and it attaches to a peptide antigen, a small fragment of a larger protein called GP100, which is a protein manufactured by melanocytes of the skin and by melanoma cells. So tebentafusp is the first ever ImmTAC to be approved as a cancer treatment. Now, this does all get a bit confusing, so I thought I would try and compare and contrast the bispecific T cell engagers where there's no FC region, the bispecific T cell engagers or bispecific antibodies where you do have the back end of an antibody for that increased lifespan in the body, and then the ImmTACs where it's the tip of an antibody fused to the tip of a T cell receptor. With the two antibody-based treatments, they use the tip of an antibody to attach to something on the surface of cancer cells. With the ImmTAC, it's the T cell receptor bit that attaches to the target on cancer cells. And as I've said, the advantage with an ImmTAC is that you have a much wider range of potential targets to choose from, rather than the BiTEs, where your target has to be something naturally found on the surface of the person's cancer cells. Also, with the ImmTACs, you get the increased sensitivity of T cell receptors. T cell receptors only need a tiny amount of their target to be presented to them in order for engagement and the activation of T cells to occur. With the BiTEs, you need a lot more of the target to be present in order for the T cells to get engaged. But the ImmTACs, one of the limitations of the ImmTACs is you have to do HLA matching, because the T cell receptor you've chosen will only attach to its target if it's presented to it by a certain MHC protein. And as I've said, there's lots of diversity in the human population as to which MHC proteins our cells have on their surface. So you have to do HLA matching, because the HLA genes are the genes for making the MHC proteins. With all of them, they tend to be given as in repeated treatment cycles, but the BiTEs that don't have an FC region, the treatment cycle is a continuous infusion. Whereas if you've got an FC region, it can be much less frequent. And even the ImmTAC, it looks like you can get away not with a continuous infusion, but maybe a weekly infusion. The ImmTAC seems to stay stuck to its target and not get destroyed at the same rate as the BiTEs. The BITEs, both those that do and don't include an FC region, are mostly in trials for the hematological cancers, leukemias, and lymphomas. The IMTACs are mostly in trials for solid tumors because you struggle to find things on the surface of solid tumor cells that aren't there on crucial organs and tissues that you can't afford to attack and destroy. So the BITEs often cause really toxic side effects. For people with solid tumors, the IMTACs look better. You've got a better chance of finding a target that is exclusive to the person's cancer cells. The side effects of these treatments are similar to the side effects of CAR T-cell therapy: cytokine release syndrome, neurological toxicities, increased risk of infection. You've got a lot of cancer cells being destroyed very, very quickly, and that can cause the immune system to really quickly get out of hand, and just masses and masses of cytokines being released by various different white blood cells that then cause massive drop in blood pressure and high fever. It can be life-threatening. So you have to be careful with the administration of these treatments. Often the dose is gradually ramped up over a period of 2 or 3 weeks to monitor the patient for side effects and treat them as they occur, so that you can gradually reach the therapeutic dose that you want to give, but in a safer way than just going straight in with that high dose. And then final group of treatments I want to mention are the CAR T-cell therapies that I did mention last week, but this week it's all about the innovation and improvement. So brief description of CAR T-cell therapy before I look at the innovations. So CAR T-cell therapy is when you take some of the person's own T cells, you insert into those T cells a new gene, a gene for making an antibody-based protein that would never normally exist in nature, a protein called a chimeric antigen receptor. The tip of a chimeric antigen receptor, the tip of a CAR protein, is taken from an antibody. So like natural antibodies, they attach to intact proteins found on the surface of cells, and your challenge is to find proteins on the surface of cancer cells that it is safe enough to target, because these CAR T cells are very active, they attach to their target, and they quickly kill any cell that they have become attached to. This is the structure of the CAR protein. You've got the tip of an antibody, that's the binding domain. There's a hinge, there's part that spans the outer membrane of the T cell, and then you've got the internal activation and signaling domains. At the moment, CAR T-cell therapies are NHS-approved treatments for some people with hematological cancers. Mostly the B-cell cancers like acute lymphoblastic leukemia, B-cell non-Hodgkin lymphoma, you see destruction of healthy B cells as well as cancer B cells, and so the person's ability to produce antibodies to fight infections is impaired because you've killed off the healthy antibody-producing B cells as well as the unhealthy cancer-causing B cells. We use these on the NHS to treat people with acute lymphoblastic leukemia, B-cell non-Hodgkin lymphoma. There are a couple that are licensed as treatments for myeloma, but NICE has not made a decision on those yet. The reason why CAR T-cell therapy is an effective option for people with hematological cancers, it's about these 4 things that I've listed on these tables. You've got available targets, things that are safe to target because the healthy counterparts of the cancer cells are not necessary for life. You can afford to destroy all the person's B cells as well as their cancerous B cells, because not having any B cells doesn't kill you. Often those targets are also very abundant, and you need quite a lot of the target to be present in order for the CAR T cell to be engaged and to kill its target. Another useful feature of hematological cancers is that the cancer cells are naturally in places where T cells go. So your CAR T cells find the cancer cells pretty easily, and those cancer cells are not embedded in the very immune suppressing environments that you find in solid tumors. So these CAR T cells can remain active and kill lots of cancer cells. The reasons why we don't have CAR T-cell therapies against solid tumors is that none of those 4 features exist with the solid tumors. It's very difficult to identify safe targets for the solid tumors. Anything that's present on the cancer cells is likely to also be present on their healthy cell equivalents that you can't afford to destroy. In clinical trials of CAR T-cell therapy for solid tumors, there's usually a handful of deaths in every trial because you've got the lethal destruction of healthy cells. Also, the targets tend to be more patchy. There's a lot of heterogeneity, a lot of variation in the cancer cells of a solid tumor. So some cells will have the target that you're going after, some won't. So you might get some cancer cells destroyed, but others not. Another problem is that the cancer cells aren't in places where T cells naturally go. T cells go to lymph nodes, they go to lymph tissues, they go to the bone marrow, they're in the blood. They are not necessarily traveling in and out of tumors. And even if they get inside tumors, they meet a very immune suppressing environment, lots of regulatory T cells, lots of myeloid-derived suppressor cells, lots of tumor-associated macrophages, lots of cells that quickly suppress or even destroy the CAR T cells that you gave as your treatment. So there's many obstacles against having effective CAR T cell therapies for people with solid tumors. Now, how to overcome these problems? Well, there's a myriad of different approaches being tried. I've listed just some of them here, but honestly, the amount of innovation, the number of ideas just goes on and on. Using intratumoral injections, using CAR T cells with multiple targets, combining CAR T cells with other forms of treatment, creating CAR T cells that produce a CAR protein and a T cell engager protein, or creating CAR T cells that also produce virus proteins to try and trigger a more powerful immune response. Loads and loads of different ideas being tried. And I thought it might be interesting just to show you a few of the trials going on around the world. Now, most of the innovation in CAR T cell therapy is happening in China and America. So very, very few trials occurring in Europe, even fewer in the UK. Most are probably 60% in China, 35% in America, the remaining 5% in Europe and Australasia, I would say. So you're not going to be able to recruit patients into these trials, but I thought it was still interesting to see what sorts of things are being used as the target. This is in glioblastoma, a lethal form of brain tumor, very, very difficult cancer to treat. And you tend to find that the CAR T cell therapy trials in solid tumors, either they are being run in people with a cancer type where there's a particularly good looking target, or they're being run in a cancer type known to be sensitive to immunotherapy and therefore seems promising to use CAR T cell therapy, or a cancer like glioblastoma where we've made so little progress and have so little to offer that the bar is set very low as to what success looks like. So these are just some of the trials involving people with glioblastoma. Another set of trials, this time trials in people with non-small cell lung cancer. And again, I've tried to pick out trials where there's some kind of innovation going on of different sites of delivery. CAR T cells will also produce signaling proteins like interleukins. CAR T cell therapy co-administered with a checkpoint inhibitor and chemotherapy. Lots of different ideas being tested out. As I've said, not so much in trials in Europe, but mostly in China and America. People are also testing out CAR NK cells rather than CAR T cells. NK cells, natural killer cells, do have some capacity to kill cancer cells naturally. They're rather rare inside tumors, and they don't live for very long in the body, which kind of works against them. But because they naturally do have a capacity to kill cancer cells, and because you don't have to do tissue matching, you don't have any risk of graft versus host disease, they're considered to be pretty safe. So there's various CAR NK cells being developed and CAR macrophage cells. Macrophages, one of the big advantages of CAR macrophage cells is that macrophages do make their way into tumors. They don't seem to get suppressed and inactivated by the immune microenvironment. So they are going to the places where cancer cells naturally live and remaining active. But it's still very early days in the development of CAR macrophage cells. So that's a long road ahead. And then lastly, TCR engineered T cells. This is where instead of inserting the gene for a CAR protein, you insert the gene for a novel T cell receptor, a T cell receptor that you have chosen because it targets a peptide antigen which you think is going to be present in the MHC cups on the person's cancer cells. The TCR engineered T cells, there are a lot in trials, but again, nothing licensed yet and probably still quite a long road ahead. So I hope that gives you a flavour of both why we use antibodies as cancer treatments and why there's a bit of confusion over whether these treatments are targeted treatments or whether they're immunotherapies, that it all depends on how it's been designed and what the target is. Lots of different mechanisms of action depending on how it's been designed, and lots of improvements being made as well. Lots of tweaks and changes and new ideas being tested out, lots of clinical trials with these novel therapies, and lots of progress being made. I would say particularly with the antibody-drug conjugates, there's a lot of those in trials, and with the T-cell engagers. Novel designs such as those that include an FC receptor, the IMTACs, where you're broadening the range of potential targets by using the tip of a T-cell receptor rather than a tip of the antibody. That is expanding the possibilities of how we might use these treatments and opening up the possibility of using them as treatments for solid tumors rather than just as treatments for hematological cancers. Okay, I'm going to stop there and I shall hopefully stop sharing. Oh yeah, stop sharing my screen.
Has anybody got any questions? If you want, you can write them in the chat box as well. But thanks, Elaine. That was absolutely brilliant. I was a bit worried coming back after the summer and thinking to myself, will my brain be able to take all the science? But actually, you've simplified it, and it makes... For those of you who don't know me, I've been giving forms of chemotherapy, systemic anti-cancer therapies for the last 30 years. And I've watched some of these developments happen, and I've taken, you know, when I was a research nurse, some of the trials working in the day unit, I've seen drugs go through the late phase trials and now become standard NHS treatment. And it's just really what's come out in the last 10 years. It's so exciting. And my question was actually going to be, is there anything for gliomas, glioblastomas? And of course you answered that. And I think I sort of feel a bit want to have a little breathlessness attack thinking about the trastuzumab deruxtecan, and if NICE approve
I
it.
really don't know what the NHS is going to do with that.
No, but anyway, we will rise to that challenge as and when should it happen. And I think, like, when it comes to some of the, like when you were talking about the T-cell engagers, I was thinking there, you know, with all the drugs that are going through the trials, and we do have some research nurses, even if some of them are only new to the late phase research team, but that is one of the things. And I'm just thinking more recently, NICE did approve, it was for squamous cell carcinoma, but it was cemiplimab. I can never, until I've practiced pronouncing it. And they approved that on the back of phase 3 trials that actually showed it worked, and there was nothing else for patients at that particular time. So that's where they brought it in under the Cancer Drug Funding Initiative as well. So for me personally, that has been a beautiful summary of all these new drugs that have come out. And thank you, Elaine. Has anybody got any questions?
I think all these treatments do bring their own challenges in administration and funding, and you look at the sheer number of them in trials at the moment and just think, I don't know how you pick a winner. It's often difficult to predict which ones will stand the test of time, which ones, and they don't always fit easily into this move towards precision cancer medicine. Like, it sounds so good, precision cancer medicine. I think as human beings, we love the sound of it. It sounds precise, it sounds free of side effects, it sounds effective. But a lot of things like the antibody-drug conjugates, there's no requirement for biomarker testing. You presume the target's going to be present, you give the treatment, and it doesn't really fit under that. I think it's a difficult one for the NHS. Like, I was looking at the Ascend career pathway work done by NHS England and collaborators of defining what the cancer workforce should know at each, what cancer nurses and their colleagues, what knowledge should be embedded in their brains at each stage of their journey as they go up through the rungs of the ladder of the banding system. And you look at what they've said in terms of the knowledge needed by each set of nurses at each band level. And there's a lot of talk of cancer biology, but also precision cancer medicine. I think it's just become this phrase that everybody's latched onto. It sounds so good, and yet it becomes a bit of a misnomer in a way with some of these antibody-drug conjugates, in that we are not going to be using them in a very precise way.
Yeah.
There's no requirement for biomarker testing.
Yeah.
And yet they are useful treatments. It's not to do them any disservice. And some of the ones that fit into that category of precision medicine are not necessarily great treatments. Just because they fit into that nice category that sounds so good doesn't mean they're any better.
Yeah.
That's always the way with cancer treatments. Sometimes blunt tools are good, sometimes precise tools have their weaknesses.
Yeah.
And you can't just make generalizations and presume that greater precision is necessarily the better way forward.
Well, and if we go back to your first talk, you talk about how cancer cells are clever and how they will adapt and move around and circumnavigate. So some of the things that we will develop, they'll find ways to move around or develop.
And that's when precision is not our friend. Because the more precise you get, the more you're selectively killing small populations of cancer cells and leaving others behind. Sometimes you don't want precision because there's diversity. So sometimes precision just sounds better than it can deliver, I think.
And it's also remembering that precision doesn't mean it has any less side effects than our old standard treatment. I think over the years, what we've also learned with the precision drugs, or the more targeted drugs, they can have side effects that can be just as detrimental to a patient as the old-fashioned chemotherapy. Yeah,
I think it's wrong to think of them as targeting cancer cells. We're targeting something that we believe is more important to cancer cells or is more prevalent in cancer cells than in the rest of the body, but it will not be completely absent or completely unused by healthy cells. It will produce side effects, as you say.
Shuba has just said a thank you for presenting a complicated topic in a pleasant way, and it is. It was very good. For
Cool.
those
Well,
of you
that's
that have,
helpful.
I've taken all your names, and I'll send you on a copy of all the slides.
Yeah.
Because even
fantastic.
I found that the detail in some of the slides was very useful to have. Sorry, Shuba, did you have a question? What is it? Is ADC is applicable to oral meds too? Yes, it would be. Yeah. ADC, I'm just trying to think, have I missed something?
Antibody-drug conjugates.
Oh yeah. Have we got it would be, wouldn't it, if we have any in development?
I don't quite understand the question. What does, can you explain, does the question, is it more even?
Yeah, Elena. So I don't know whether my question is really like does makes any sense, but there was just a small query. So we're talking about the antigen drug conjugate. So the more focus was given on the IV medication. So I was just thinking whether it is applicable to the oral chemo medicines also, or does it have a different way of like antigen antibody combination things and all?
Or I don't know if this answers your question or not, because I'm still struggling to understand. But one thing to bear in mind with all of these antibody-based treatments is none of them can be given orally. Is that what you're asking? Like, they are all protein-based.
Yeah,
antibodies are protein.
I got the answer now. Yeah, I got the answer now. So
Because
we
they
do
are
have chemo
proteins.
meds, yeah, oral meds. So I was just thinking, does this applicable for oral chemo drugs also? Like, no. So I got the answer is
All the
no.
antibody-based treatments are proteins, and if you gave them orally, they would be destroyed by your stomach acids and by your digestive enzymes, because that's what, you know, your body's very good at digesting proteins to release their amino acids, so they'd be completely destroyed by your digestive tract. So you have to get them straight into the bloodstream or straight into a tissue. You can't give them orally.
Okay, thank you.
Great. Well, thank you, Elaine, once again. And just to let everybody know, the next session is on the 10th of October, followed by another one on the 17th of October. I'll send you out the details, but keep those dates in your diary. 6 o'clock, MS Teams, or there may be more local groups in your own hospitals that will get together. But the MS Teams format, it seems to be working well. So once again, oh, Elaine, I had just one quick question. Do you have any information about the safety issues around the handling of any of these treatments? You know, in the preparation and the actual handling, administering. So safety of staff handling these. Don't have to answer that question now.
Not offhand, but I can have a think. Sometimes ESMO produces useful guidance.
Yeah.
And Clinical Care Options website is a source of great information as well. I'll have a look.
Yeah, it's just more like, has there anything come out recently that might make the handling from the safety of the personnel handling the drugs, is it any different to what we've done when it comes to handling general cytotoxics, the whole lot? So if you have come across any information, please let me know. That would be great.
Okay.
Please.
Yeah, absolutely. I'll have a look.
Thank you. Thank you, Elaine, and thank you everybody for joining. Speak soon.
Cheers.
Bye.
Thank you so much, both of you. Thank you. Take care. Bye. Bye.